Welcome again, and thank you for joining us today. My name is Gill, and I work as a licensing associate with the Office of Technology Licensing at Princeton University. I welcome you all now to the Faculty Innovators portion of Celebrate Princeton Innovation. For the overall schedule, we have two sequential tracks. The first focusing on life sciences, and the second focusing on tech. Each track will feature five faculty innovators who will each be giving a short 8-minute presentation. At the end of each presentation, we will have about 2 minutes of Q&A. So please input your questions in the Q&A tab on your screen during the presentation so we can bring them up at the end of the presentation. You will also have the option to upvote existing questions asked by your fellow audience members. We will try to get through as many of your questions, but if we run out of time as we have a busy schedule, please feel free to reach out to our team at the Office of Technology Licensing. With that, I will not take up any more of your time, as I'm sure you're excited to hear more about the cutting edge innovation happening at Princeton. So let's start. I now welcome our first speaker in the life sciences track, Professor Joshua Rabinowitz. Professor Rabinowitz is a professor of chemistry at the Louis Sigler for Integrative Genomics, and he will be presenting on therapy that robs cancer of its growth potential. Welcome, Josh.
Thanks so much for the introduction. I'm going to be telling a story that I particularly like because it's a very Princeton story. And I say that because the way that we're robbing cancer of its growth potential is by targeting a particular metabolic process, folate metabolism, that was targeted by a wonderful chemistry professor here, Ted Taylor. And Ted Taylor made the molecule that you see on the screen here, pemetrexed. And I have to say, there have been several very exciting events in the chemistry department at Princeton over the past 50 years. Most recently, of course, the Nobel Prize to Dave McMillan. But this certainly stands out as one of the seminal events in the chemistry department. And this molecule has now helped millions of cancer patients worldwide, as well as leading to wonderful developments at Princeton, like the Frick Chemistry Lab. And so that molecule works on the process of folate metabolism, as does the very first rational agent for targeting cancer, made by Sidney Farber, who ended up with its name accordingly on the Dana-Farber Cancer Institute in Boston. And so folate metabolism is a really central process in cancer and in enabling cancer growth. And it's one that we thought we could learn more about by applying new technologies that we developed in the lab involving liquid chromatography and mass spectrometry to study exactly how these metabolic reactions work. And through that effort, we identified a critical step being the loading of the one carbon unit that this cofactor is eventually going to put into DNA precursors, adenine, guanine, and thymidine, from the amino acid serine, as shown on the screen here. And so we were wondering if we could cut off that loading of the one carbon unit as a new way to target cancer. To get at that, we wanted to go after one of the most upregulated genes in all of human cancer, the SHMT2 gene, which carries out this reaction. However, when we did that, you see that we only got a modest effect on tumor growth. And so, unfortunately, this would still be clinically classified as progressive disease. And so we needed to do better. And we were able to use our mass spectrometry strategies to figure out how the cancer was escaping inhibition of this highly upregulated enzyme. And it was doing it by using a very similar enzyme that sits in the cytosol rather than mitochondria of the cells, such as SHMT1. And when we were able to target both enzymes genetically, then we could make tumors disappear. And so on one hand, this was exciting because through using metabolic and chemical approaches, we identified targets that are redundant, so it would have been hard to find in a genetic screen. However, at the same time, you say, what's the value of finding two targets that you have to get together to treat cancer? Well, the good news here is these targets had very similar active sites to carry out the same reaction. And we were actually able, working with Han Kim, whose picture I'll show you on the next slide, to find a family of small molecules initially developed as herbicides that simultaneously target both of these enzymes in a stereoselective manner. We're able to establish crystallography for the small molecule inhibitors bound to the enzyme and use that to get to an optimized small molecule with promising pharmacological properties. And this is the beautiful molecule made by members of my lab in collaboration with Han, who really was the intellectual driver of this effort. And we're so lucky to have him as part of the innovation ecosystem here for drug development at Princeton. What does this molecule do? Well, to get at that, we were fortunate to be able to collaborate with our colleagues at the Rutgers Cancer Institute in New Jersey, and particularly Danny Arons, who has tremendous models for leukemia, in particular T-acute leukemia, which is a disease that's treated with an antifolate as standard of care currently, methotrexate. And we're able to show that our molecules had single agent activity in this disease in vivo with meaningful lifespan extensions in terms of slowing death from cancer. Because methotrexate is already used to treat this disease, we're very curious if as cells develop resistance to methotrexate, they would also lose their responsiveness to our new therapy. And what we found that really got us excited is that exactly the converse happens. As cancer cells become resistant to methotrexate, they do this through changes in their metabolic pathways that end up making them about 10 times more sensitive to our therapy. So all of a sudden, we're ideally situated to work together with methotrexate or after methotrexate in patients who have failed standard of care therapy and have nothing else to turn to. And indeed, we were able to show in these same animal models that we have synergy in vivo with methotrexate and now getting quite meaningful extensions in lifespan. We didn't stop with these animal models of cancer. We also wanted to go forward to use the most clinically predictive models, which are patient-derived xenografts. And again, you see strong activity of our new SHMT inhibitor and also combination activity with methotrexate. And based on the strength of this data, this program was out-licensed to Raphael Pharmaceuticals, who's very actively pushing it forward towards the clinic. Hopefully within 18 months, we'll have initial human testing to report. Now that's been out licensed, but certainly didn't stop our research efforts here at Princeton wanting to do yet better. And I'm happy to say that together with Zemer Gattai, who's really been the leader of this program, we've identified other targets within folate metabolism that can also be hit by interesting small molecules where Princeton has patent protection that seem to show even better single agent activity against T-acute leukemia. And so if you're interested in this, this is an open licensing opportunity that Zemer or I would be delighted to discuss with you. Now, in the minute or two that I have remaining, I want to point out how by making these molecules, we really don't just enable treating the disease we initially went after, we make biological tools that can teach us about other diseases. And in this case, I'm going to talk about a very prevalent disease, fatty liver disease. This affects about 1/3 of US adults and is becoming the leading cause of liver failure in developed countries, including the US. And a major driver of fatty liver disease is the fact that when we eat sweets, or even worse, drink sweets, then our liver turns them into fat, stores that fat in the liver, and this causes liver failure. And you can see this here in a mouse model. We give the mouse the equivalent of Coke, and you see that it really turns on their synthesis of fat. That's the liver DNL flux. Okay, that's the liver making fat from carbohydrates and then accumulating fat in the liver. And through very interesting biology that we're only partially understanding so far, we've been able to find with these small molecule inhibitors that we can turn on and turn off this sugar-induced production of fat in the liver by manipulating the SHMT system. And we can use these small molecule inhibitors that we initially made for cancer to turn off liver fat synthesis and prevent, at least in mouse models, fatty liver disease. And this is another exciting opportunity that Raphael will pursue with these new family of small molecules. And so with that, I want to thank you for your attention, thank my lab group and funders, and take any questions.
Thank you, Josh, for that insightful presentation. We have actually a question from our audience. Could you please talk to us about the current stage of development of this technology, and what do you think the most promising indication may be?
Yeah, I think we're very interested in getting initial clinical proof of concept in hematological malignancies, in particularly T acute leukemia. This is probably the malignancy that's most sensitive to methotrexate, and this technology is complementary to methotrexate. And the same cancer cells that are sensitive to methotrexate are often sensitive to this inhibitor. And as I say, we're getting towards optimization of a clinical candidate that could go into formal development studies for that. That said, I think the biggest long-term potential is in metabolic syndrome and fatty liver. And you saw the very interesting data that we have there. And we actually have kind of three complementary mechanisms that all seem to converge to mitigate fatty liver disease. That's earlier. That's going to be a harder road to clinical proof of concept. They're much more expensive and long-term trials, but I think it's a huge unmet need in the United States. It's something that we are very optimistic we can contribute to helping with.
Perfect. Thank you very much, Josh. So now I welcome Professor Daniel Cohen. Professor Cohen is an assistant professor of mechanical and aerospace engineering and will be presenting on faster healing with bioelectricity. Welcome, Daniel.
Thanks. It's great to be here. I appreciate it and appreciate your time because you're about to be subjected to a whirlwind tour of what my lab's been up to for the last 3 years since we opened. So I'm Daniel. I'm an assistant professor of mechanical and aerospace engineering. And one of the key areas we're working in is to try to understand how to use external cues to dynamically accelerate and improve wound healing. So just so that we're on the same page and everyone understands what I mean by healing, this is a beautiful movie from the lab on actual skin healing over 2 days. And so this is mouse skin. And what you can see here is purple dots that are individual mouse cells moving over the top of a green layer, which are the lower levels of skin. So purple is what you touch sort of on your skin, and green is below that. And this process together produces a beautifully choreographed 3D healing approach that allows us to spontaneously heal in a self-organized way. It's elegant. It's important. It's part of the reason why multicellular life can exist, that we can self-heal, but it's not perfect and it has issues. And 5% of healthcare costs globally today are related to treating acute and chronic wounds because wounds don't always heal the way we want them to. As we get older, they heal more slowly. Diseases like diabetes can cause wounds not to heal at all, producing ulcers. Even if something does heal, it can produce significant scarring that can be debilitating in its own right. So what we're trying to do is come up with new tools to actively program and enhance wound healing. And because my lab specializes in swarm dynamics and collective behavior, when we look at a process like this with this beautiful coordinated crowd type motion of cells, what we thought was, can we herd these cells? Can we take advantage of existing paradigms in kind of swarm control and crowd dynamics to come up with a general approach to make wound healing more effective? In other words, can we take tissue that might look like this in a Petri dish and do this to it? Sheep herding, our oldest swarm technology. It's 7,500 years old or so. It's a beautifully elegant process where a farmer and a few dogs can move some sheep around just by having that right perturbation response, knowing how the sheep respond to the dog. So we want to come up with a sheepdog for living tissues. So there are two constraints we had. First, we want to be able to program this dynamically. We don't just want to put something in and set and forget it. We want to be able to look at tissue, intelligently produce a particular growth and motion response, and adapt that over time. And second, we want this to be broadly applicable. We're not looking for a sheepdog that requires you to genetically modify the patient, for instance, to respond to something like an optogenetic cue. And so when we started to think about the possible sheepdogs that are eligible here, the first thing we evaluated were chemical cues, which are important for sure, but we couldn't actually find one that would effectively control migration and growth in healing. And they're very hard to program as well spatially. Mechanical cues are also important, but we don't know enough to dynamically kind of program those and shape them in a therapeutic way yet. And so we got to electrical cues, which are something that as a community, we're very good at programming precisely. But you have a couple of issues, one of which is there's a natural pop cultural stigma around non-neural bioelectricity. And if any of you were thinking about Frankenstein, you're on the right track. there is a almost comically bad history of quack medicine in this field, and I give a lot of nightclub talks just about that problem. But what's really interesting is all of this pop cultural reference hides some of the real science that could really be helpful for biomedical applications in the future. So for instance, almost 200 years ago, Emile Dubois-Reymond, who wrote the first textbook on electrophysiology and was famous for performing experiments shirtless, cut his thumb and measured a current in the injury site. And there's nothing mystical about this. We're basically a collection of tubes full of salt water. If you prick us, we leak. And if we leak, ions are moving. And if ions are moving, you get a current. And effectively, you get an electric field of about 1 volt per centimeter in things like skin wounds, where it's positive on the outside, negative on the inside. Now, all of that's great, except that it's really important when you consider that cells can sense and follow these natural ionic fields in a process called electrotaxis that's been shown to help everything from embryonic development to wound healing, as well as being implicated in cancer growth. And these electric fields actually tell cells where to go. It's a navigational cue, and they can increase the speed of motion. So great, we can make cells move where we want faster. Why don't we have an electric Band-Aid today? There's not a short answer to this, but the quick version I'll give you is that most of the work in the field is focused on single cells, which is great for understanding molecular mechanism, but it's hard to translate. Then there's also been a lot of work on just direct in vivo tests on rodent models using various electrode configurations, but it's such a complicated development cycle, and it's so hard to get information out of those systems that no existing device is particularly effective right now. There are 2 approved commercial products, the PosEffect ring electrode system designed for diabetic ulcers and the Procellera electrode grid system that produces tiny fields that disrupt bacterial growth. Neither is a clinical standard of care, and neither in meta-studies is necessarily better than a control. So what I think is missing in the field, and what my lab's been working on, is targeting that white space of using bridge models that have been so effective in pharmaceutical and biomaterials design to look at organ on a chip or organoids, or just more organotypic models that allow us to do high throughput screening of optimum stimulation that might allow us to more effectively engineer and smartly heal injuries. So let me give you a tiny bit of data on how this looks and how we've gone about it. So taking this approach of using the sheepdog based on electric cues, the first thing we needed to prove was that you could even herd a patch of tissue, because that wasn't really clear at the time. So the first thing we demonstrated was that, yes, in fact, you can take this patch of, say, engineered kidney tissue and just tell it to move to the right electrically. And that's pretty interesting. You can even make it dance back and forth if you just switch the field direction. So our first studies were basically one-dimensional proof of concepts. And when the lab opened, what we wanted to do was to really test the boundaries of how much you could really program cells using this. How powerful are these electrical cues? And so what we did was build a device called Sheepdog with a ridiculous acronym that describes essentially a multi-electrode bioreactor that allows us to independently address and program multiple electrochemical electrodes to produce any arbitrary 2D electric field that then be applied to a living tissue. And using this device, we did a demonstration where we took cells from mouse skin. Each little dot here is a cell, is the nucleus, and we told them to move in a circle. Absolutely not what you want to do in a real wound, but it really demonstrates how responsive tissues are to these simple electrical cues. This white arrow shows the direction the field is pointing at any given time. So we can really program this. And since then, we started to fine-tune things. We worked with more complex tissue. So this is a layer of mouse skin that's much more mature, kind of like the upper layers of your skin. Still a single layer, but it's hard to work with. But using a combination of field stimulation and some drugs that enhance cellular sensitivity to field stimulation, we were able to translate this whole patch of skin at millimeter distances across a Petri dish in real time. And that's something we'd never seen before, and it was really promising. So what we started to do then was to mimic the geometry of real wound fields and to produce something that would actually produce a convergent healing process. So here we're telling this wound to heal faster by moving inwards to the center. And so we can more than double the speed of healing, again, in an in vitro Petri dish context, but it's quite promising so far. So what we'd like to do now is to build this into these bridge models using things like human skin equivalents and complex 3D tissues that we can actually much better get a sense of the parameters, so that we could imagine a farm of electric Band-Aids being tested on an array of little mini tissues, really helping us explore the optimal stimulation space to produce the first truly smart Band-Aid that can measure your wounds and stimulate back in intelligently. We need some help connecting to these types of model systems and some of the expertise in the skin industry, as well as thinking about different ways to do these integration loops. But we think it's promising and got a lot of potential, and we're excited to move forward. So thanks for your time. And a shout out to my groups and the funders at the National Institutes of Health and NSF. And I'll take questions.
Thank you very much, Daniel, for that exciting presentation. We actually have a question from our audience. So could you talk to us about some of the challenges or unanswered questions in commercializing a product that may use this technology?
Almost all the work in this field has been done in rodent models so far, and the mechanics of rodent skin are very different from human skin. So that's part of the reason why we really want to test this in a human skin context using these organ-on-a-chip models. Another detail is almost all the work's been done in 2D. So we're really excited to try these much more realistic 3D systems where things are probably going to behave a little differently. And we think that's where we need to spend time optimizing.
Thank you so much, Daniel. Next, it is my pleasure to introduce you to Professor Mohamed Donia. Professor Donia is an associate professor of molecular biology and will be presenting about a method for screening the gut microbiome's effect on drug metabolism safety. Welcome, Mohamed.
Thank you very much for the introduction and for the invitation to speak today. I'm very excited to tell you about the work we've been doing in my lab for the last few years. So I'll just jump right in and motivate this talk by telling you that, as many of you know, drugs are really developed with the population in mind, meaning that we do large clinical trials with many patients, and as long as the drug works for most patients, we're good to go. We approve this drug and it gets into the actual clinical use. But this does not take into account individual differences in response to the medication, leading into different variabilities and disparities in the way different people respond, or different patients with the same disease respond to the same medication, whether in therapeutic efficacy or with toxicities that are related to the use of that particular medication. So if we look at the human genome now, the human genome can definitely explain some of those variabilities, but not all. So maybe half of those variabilities can be explained by differences in the human genomes, but not all. So what are the other sources of variabilities that exist in the drug use or in therapeutic efficacies and toxicities? And today I would like to give you a little bit of a rundown of what we've been doing in my lab, basically looking at our gut microbiome. So those are the hundreds of species that exist of bacteria, fungi, viruses, and protists that exist in our guts all the time. They have lived with us for many, many years, and we cannot live without them. Those are very, very active microbes, also active biochemically as well. So estimates now say that there are hundreds of thousands of uncharacterized enzymes and biochemical pathways that actually are encoded by the gut microbiome. So we asked this question of, could this be one of the forgotten organs in the human body that could explain some of the variability that exists when we use therapeutic agents in patients? And could the microbiome be playing a role in this? And this is not, we can't claim that this is our invention. This observation has been known for many, many years, almost 100 years now, where certain drugs, about 50 in the most case, have been correlated somewhat to the gut microbiome as being directly metabolized or affected by members of the gut microbiome, especially bacterial members. So there are many ways that the gut microbiome can do this. And in the known cases, there are cases where we take an orally administered medication, it goes into the gut, it gets metabolized by gut bacteria into, let's say, a toxic metabolite, or it gets degraded completely into an inactive metabolite. And then those metabolites can have a subsequent effect. If it's a toxic metabolite, it causes toxicity. If it's an inactive metabolite, then most of the dose is gone. So this is one area. Another area is even non-orally administered medications by injection, for example, can end up reaching the intestine through biliary secretion, which can also lead into interactions with the microbiome that can lead into adverse effects or different therapeutic responses. So this has been known for many years, but what has not been known or done is really a good standardized method of addressing this in the clinic or in clinical trials. So the way people have been addressing this before our work has been to basically take the gut microbiome from a single person, which could have in it hundreds of species of bacteria, culture as many of those as you can, and then test each of those individual microbes against a few drugs and see if they actually can metabolize those drugs, can perform biochemical transformations on them, and then evaluate the drug metabolism by the microbiome this way. What we have decided to develop a few years ago is a whole community assay, basically taking the entire gut microbiome and understanding its entire biochemical repertoire or biochemical capacity to metabolize hundreds and hundreds of drugs. So the advantage of this, there are several advantages to this method. We can look at multi-organismal contributions. For example, one microbe can metabolize a drug, another one can revert that metabolism or metabolize it even further into a different metabolite, as well as this can allow us to finally look at inter-individual variability. It's not feasible to take a gut microbiome sample from 100 different patients, isolate thousands of species from each patient, and then test each individually against certain drugs. And this allowed us really to do some systematic mapping of drug metabolism by the microbiome. So I'm going to walk you through what we decided to do a few years ago and was recently published. This was done by two amazing students in the lab, an MD PhD student, Bahar, and a quantitative and computational biology student, Jaime. So basically what we decided to do is to take 20 healthy donors, mostly students from Princeton, that gave us microbiome samples. And then we developed a method to culture the gut microbiome samples. These are stool samples. We can culture them in the lab in high throughput and then measure how much recapitulation of the microbiome can we have in a test tube. But also, not only that, but we can also measure really the personalization of these cultures. So if we take a culture from donor 1 versus donor 5, do they really represent the individualism that happens in their microbiome or the personalization in their microbiome in their actual fecal samples? Or are we culturing always the same things? So this is a very busy slide, but it definitely shows that basically each box here is one of our 20 donors. And then each column is a different culture that we've done from each of those donors. And the very far left one is basically the fecal sample. So I hope you can appreciate this, but there are essences of personalization here, where, for example, this group of bacteria exists only in donor 6 and not in donor 7. And same thing here in donor 12, and there's some group of bacteria that exist, but not in donor 13, and so on. So we can recapitulate this personalization aspect in the cultures that we can maintain in the lab and cryogenically preserve, which is very important, so we can repeat the assays over time. So once we've developed this method to basically culture the gut microbiome in aggregate as a very complex community, personalized community, in a test tube from each individual, we went ahead and we tested hundreds of orally administered, clinically approved drugs in a screen, in a biochemical screen that we designed. So basically incubating those drugs with the microbiome and seeing whether we can observe metabolism or not. And we were really shocked to see that 13% of the drugs that we tested were heavily modified by the microbiome. And those drugs span all types of pharmacological classes, all the way from antibiotics to antidepressants, non-steroidals, and others. So this was the initial result. We then started looking individually at those drugs, and we were really surprised to find that in several cases, we found metabolites that actually were seen in humans, but have never been explained how they're made. They're not made by the liver, they're not made by the intestines, and we did not know before our study that the microbiome is the contributor of these metabolites. The last thing that we did was to measure what we set up to do at the very beginning, which is to see if we can actually quantify inter-individual variability in what we call microbiome-derived metabolism, or MDM, using these personalized communities. So we took the 20 different donors, excuse me, and we tested them against 23 selected drugs, and basically quantified what the differences between the donors in their ability to modify each of those drugs. And this is when we really saw the power of our approach. So here's just a heat map showing the 20 different donors here, and then the 23 drugs over here. And you can see 3 different patterns. Either when you see something that is completely blue, that means that the drug is completely gone in these samples. So this is all of the donors metabolize that drug completely. If we see all yellow, that means that none of the donors metabolize that drug. But the really interesting cases are those where we can see that some of the donors metabolize that drug while others did not. And this did not only appear in the actual consumption of those drugs, or but also in the identity of the resulting metabolites too. If we quantify the resulting metabolites and we look at them, we see that not only if 2 donors metabolize the same drugs, that doesn't mean that they metabolize that drug into the same metabolite. One can produce metabolite 1, while the other donor can produce metabolite 2. So there's even more variability in that aspect. So overall, what I have told you about today is that we've developed an approach, an experimental approach that allows us to collect gut microbiome samples from dozens and dozens of patients, and then culture them in a personalized manner, recapitulate the specificity, or the actual the specific species that exist in each individual, test them against libraries of drugs, and then what I haven't told you about today is that we're able actually to take this into animals and prove that what we've seen in this in vitro assay actually is recapitulated in a mammalian host, and that we can also link those back, what the observations, to enzymes and genes in the microbiome that perform these modifications. We have patented this method, and we're trying to push it forward towards clinical use. The idea is that we can do this at bedside, basically before prescribing a particular drug that is known to be variable. You would want to test the microbiome and see if that patient that you're going to respond, that you're going to prescribe that drug to, will actually have a chance of responding or not, based on microbiome metabolism. So I would like to thank all of my lab members. The team that worked on this are, as I mentioned, are Bahar and Jaime, mostly with help from many other members in the lab. And I would like to thank our funding agencies, and especially the Department of Molecular Biology at Princeton. Most of this work, 90% of this work, was funded through an innovation award from our department. And basically, at the very beginning, no one believed that this is possible. No one wanted to fund this work. But of course, Princeton believed in us and gave us the money, and we were able to reach what we have now. And I would like to thank Lori at the Office of Technology Licensing at Princeton, who has worked many hours with us on drafting our applications, and also on thinking about how we can use this method in the clinic. And thank you very much for your attention.
Thank you very much, Mohamed, for that interesting presentation. We are running short of time, so we just have time for one question. So one of the questions that came in was, how disruptive to the microbiome are common antibiotics?
Sorry, can you say it again? Sorry, I didn't hear it.
So how disruptive to the microbiome are common antibiotics?
Oh, okay. Yeah, they're very disruptive. So most of the cases when we use a broad spectrum antibiotic, it really harms your microbiome, and it takes a long time for it to revert back. So yeah, that's something we've observed, and others as well. And something that we should be aware of, and that's why we hope to, in the future, to take this into account and design more targeted antibiotics that don't disrupt the microbiome.
Perfect. Thank you so much, Mohamed. I now welcome our next speaker, Professor Andrew Leifer. Professor Leifer is an assistant professor of physics and neuroscience and will be presenting about Princeton Open Ventilation Monitor. Welcome, Andrew.
Hello, thank you for that introduction. Today I'll be talking about new technology that a team of us developed to allow clinicians to better monitor COVID patients undergoing non-invasive ventilation. All right. Let's see if my pointer will work. There we go. COVID-19 showed that there is an urgent need for gentler alternatives to invasive mechanical ventilation. So traditional mechanical ventilation is invasive. The patient undergoes anesthesia and is intubated with a kind of a plastic tube that goes down their airways. And traditional ventilation relies on expensive and sometimes scarce ventilators. So while traditional mechanical ventilation saves lives for those who need it, other gentler methods are needed to support those patients who may not yet need a mechanical ventilator, but with early treatment may be able to avoid needing one later. So for a gentler approach, clinicians have turned to a number of alternatives. Two are the high-flow nasal cannula and the newer alternative called helmet NIV for non-invasive ventilation. So both of these methods provide oxygen to the patient without anesthesia, intubation, or a ventilator machine. So high-flow nasal cannula flows air and oxygen into the nostrils. In the helmet system, air and oxygen flow into a pressurized helmet. So the helmet approach is particularly appealing because it is an enclosed system. And an enclosed system is best at delivering pressure to the lungs. So this keeps the lungs open when the patient exhales. And it provides what physicians call, or clinicians call PEEP. That's peak end expiratory pressure. That's important because one of the many things that can go awry with diseased lungs is that they stick together and they don't open all the way. PEEP keeps those lung ways open even during exhalation, and that allows the lungs to take in more oxygen and to heal. Enclosed systems are also, in principle, safer for clinicians than open air systems because, importantly, there's a particle filter that sits between the patient, where all the viral particles are, and the clinician, and that keeps the virus particles away from the clinician. Finally, in principle, an enclosed system is more amenable to monitoring the patient's respiratory profile than an open system like high-flow nasal cannula. And that's important because clinicians are used to monitoring a patient's progress in a disease by observing waveforms of airflow and pressure, which I'm showing here on a traditional mechanical ventilator. And they also monitor tidal volume and pressure, which they can use to track the lung stiffness, called compliance, and that's an important marker of lung health as well. So open air systems like high flow nasal cannula can't offer these diagnostics, but helmets should be able to. Until now, however, no system has existed to provide respiratory monitoring of patients undergoing helmet non-invasive ventilation. And there's one more point here. Monitoring isn't just a clinical nicety or a useful tool, it's actually a major safety issue with the helmet system. And here's why, if the flow through the helmet gets too low because a tube gets kinked or there's some kind of malfunction, the patient can end up rebreathing too much carbon dioxide, and that can be deadly. So the lack of monitoring has been a major impediment to wider adoption of this helmet technique. Okay, so we learned all this from our clinician colleagues at our local hospital system very early on in the pandemic, and we wanted to do something to help. So we set out to make a monitoring system for helmet non-invasive ventilation that would provide the kind of respiratory monitoring and profiling for patients in the helmet that clinicians are used to from a traditional ventilator. We also wanted to design something that would sound alarms if any problems arose. And we wanted a system that would allow a single clinician to monitor many patients simultaneously because our healthcare workers are stretched thin at the moment. And anything we did needed to be fast and inexpensive so that we could manufacture them locally, and it needed to avoid the severe supply chain disruptions of components related to ventilators because everyone in the world wanted to make ventilators just as we were doing this, not surprisingly. So we assembled a large team of faculty, staff, and students from across multiple departments at the university. And we secured funding from the Princeton's Office of the Provost, as well as support from the National Science Foundation. In the end, we built a multi-patient remote monitoring system that had monitors and sensors for flow and CO2, and that were connected to a bedside monitoring system, which is over here, and that relays information to clinicians at a remote monitoring station. This system provides clinicians with all the metrics that they're used to from a traditional ventilator, including the patient's pressure and flow waveforms, the patient's respiratory rate, how often they're taking a breath, the patient's tidal volume, or how much air the patient breathes in or out during each breath, and also information about that lung stiffness that I told you about earlier, and the level of carbon dioxide in the helmet, which is important for safety. And the clinician can set acceptable ranges for all these values, and the system will sound an alarm if anything goes out of range. And importantly, we designed the system so that it monitors up to 20 patients at one remote monitoring station at a time. So we manufactured 50 of these devices in-house during the early days of the pandemic, and we tested them on a healthy human subjects in an IRB-approved study here at Princeton. You can see some of the members of our team putting those together here. For some components that were involved in traditional ventilators, like flow elements, we designed and manufactured our own versions locally to work around supply chain crunches. Princeton University is making all the details of the design and technology associated with this project publicly available online without IP protection. That includes CAD designs, the circuit diagrams, the open source software, and detailed assembly instructions like the video shown there. There's a manuscript describing the technology, which is available online, and it's just been accepted for publication in the Journal of Medical Devices. So we manufactured a large batch of these devices within the university, but to get it all the way from bench to bedside, we need to partner with industry. And that's why we're so excited that a Brooklyn-based company, Nanotronics Health, plans to commercialize a variant of this device and shepherd it through the regulatory process. We are delighted to be providing their engineers with pro bono technical support so that these systems can reach patients as soon as possible. With that, I want to show you the actual system, which I have behind me, and then I can take a question here. So I'm just going to turn on the air. This is Covey McQuarantine. He's our test dummy. You can see the helmet inflates. Air flows in to the helmet, and then it flows out through this filter here. And we have a monitor here, and in a second, so those devices, those waveforms are being monitored on the bedside device. That's here. That's a little device here. And then this information goes to a central monitoring station, either wirelessly or over an Ethernet cable. And that's shown here. And so here, you can see the central monitoring station. Here, those pressure volume curves that you use to look at compliance. Here's pressure, volume, flow, everything that a clinician would be used to from a traditional monitoring system, traditional mechanical ventilator, including tidal volume and respiratory rate. And then I want to just point out, here is an early prototype from Brooklyn's, from the Brooklyn-based Nanotronics Health Company. We're really excited that they are going to produce these devices and try to carry them through the regulatory process. I think I'll stop there so that there's still time for questions. And you're going to have to shout because the instrument's loud.
Thank you, Andrew, so much for the live demo. Very, very fascinating. Actually, we have one question from our audience. Could you please talk to us about your experience working with a private entity to commercialize this device?
You asked about the what? Working with a private entity? Say that one more time.
Your experience working with a private entity.
Well, I would say it was very positive, but I would say we're probably not necessarily stereotypical. Nanotronics Health found us based on the information we made public. and we worked with Princeton's Office of Technology Licensing to work out an agreement so that we could provide them with technical assistance. And in our case, I think it was a little unusual. We received a lot of support from the Office of the Provost as part of Princeton University's mission in the service of humanity. And so the university is not looking to monetize this work. And so we are just excited to provide information for free to anyone who would take it so that these kinds of tools can help patients.
Thank you so much, Andrew. Next up, we have Professor Sam Wang and Henk-Jan Boele. Professor Wang is a professor of neuroscience at Princeton, and Professor Boele is a visiting research scholar at Princeton Neuroscience Institute and an assistant professor at Erasmus Medical Center, Rotterdam. They will be talking about a smartphone-based neurobehavioral testing in the blink of an eye. Welcome, Sam and Henk-Jan.
Well, thank you, Gill, and I thank Princeton Innovation for hosting us today. It's been an inspiring set of presentations, and it's been wonderful to hear Josh, Daniel, Mohamed, and Andy present all these exciting projects which showcase the brilliance and the hard work of Princeton researchers. Today, what I'd like to do is talk to you about, tell you about something very exciting that has really gotten us inspired to try to help in the domain of autism, in the clinical domain, to help people with autism and their families. And what I'd like to do is show you how we are taking basic research all the way to what we hope to be practical mobile-based technology for the diagnosis, the early diagnosis, and we hope addressing the needs of people with autism. And so with that, what I'll show you here is the idea of smartphone-based neurobehavioral testing in the blink of an eye. And this means not just doing it quickly, but it also means doing it using eye blink conditioning, which is a kind of learning that's been around for 100 years and has never really been taken to mobile technology. And so with that, let me just get going here. The core idea of Blink Lab, this is a company that Henk-Jan and I have started and has gotten the first seed round of funding. The idea here is to apply mobile technology to allow high quality neurobehavioral diagnostics to bring to families, to medical professionals, and to specialists. And the idea is to look for neurobehavioral biomarkers that may be predictive of a variety of problems that come up that are of concern to those communities, whether it be autism, which is our first use case, dyslexia, developmental disorders, attention deficit, or attention deficit hyperactivity disorders. We believe that this platform has a lot of potential for helping people in all these different domains, and furthermore with telemedicine applications. So just to orient you a little bit, some of these ideas have been around for over 100 years. There are basic neuroscientific phenomena that go by names such as pre-pulse inhibition, which is if you get a small stimulus, then maybe you don't react so strongly when you get the stimulus a second time. Eye blink learning, which is learning to blink your eye to protect itself from some stimulus. And classical conditioning, which is shown here in this image of this dog, which is working with this woman and with this man, Ivan Pavlov. And this is just some stuff that many of us learned in psychology or in neuroscience classes many years ago. These scientific advances are sometimes over 100 years old, and sometimes they've been linked with autism, which itself was defined in 1943. And now I want to fast forward and say that now we have technologies such as the iPhone and computer vision, and these technologies allow these old insights to gain new life and to be put in everybody's hands. And really, when you think about it, this technology has almost gone by, almost unappreciated. This is a newspaper ad from 30 years ago showing all the great things that you could get at Radio Shack, which include like a music player and a telephone and a radio and a computer. All of these things are now available in the palm of your hand in a device. Smartphones really had their advent a little over a decade ago. And the idea is that all this technology, which used to take up a lot of space, cost a lot of money, is now available in the palm of your hand. So what we've done is we've used this as a platform to study something as simple as eye blink technology. Now, this is a member of our research group, Caroline Young, Jung. And Caroline is going to demonstrate this kind of eye blink technology. This is the front end of an alpha version of our device. And the idea here is that the camera in the smartphone is detecting automatically the features of Caroline's face using machine vision. And what we're going to watch is her reaction to a bright light applied through the iPhone. And so actually, in this case, it's an Android device, I believe. And you can see here at the yellow line here is when the flash is going to be given. So first we have a tone, which is neutral, doesn't mean anything. No reaction. And there, the flash of light, you can see that Caroline blinked her eye at the beginning of that flash. And so this is just an example of what happens when you are naive to this. You haven't really experienced it before. After getting the tone and the flash over and over again, Caroline will eventually start to make an anticipatory blink, and that anticipatory blink is predictive that the flash of light is about to come. And the simple association of tone and a flash of light is the kind of learning that Ivan Pavlov and his students and associates studied over 100 years ago. So now after, let's see if I can get this okay, so now this is now a few dozen trials later. A few dozen trials later, now that same tone is going to evoke a different reaction. You have to watch closely. Look here at the reaction over here, and you'll see that it's a little bit different. It'll come, there'll be a protective response. So here, the tone, and now you see the blink happens a little bit in advance. And so now that blink, which at first was a reflex blink, has become a learned blink that anticipates the impending flash of light. Now, this is a simple form of association. You may ask, what does this have to do with autism? It turns out that leading theories of autism suggest the idea that autism arises from difficulties in processing sensory information. And difficulties in forming these kinds of associations have been observed for decades. And what we would like to do is take that fundamental insight that sensory processing may be a problem that leads to autism and use it as an early diagnostic to predict when a kid, when a toddler or even an infant may in fact benefit from further attention. So the idea is that the cerebellum is a region of the brain that is fundamentally involved in eye blink conditioning. It turns out that cerebellar injury is also a major risk elevator for autism, and it increases the risk of autism something like 36-fold. And it's the only known environmental risk factor that is on a par with genetic involvement. So fundamentally, autism is known to be heritable. It's known to have a genetic component, and cerebellar injury at birth is increases the risk over 30-fold, which suggests that maybe the cerebellum is critically involved. And I've just shown you a kind of learning, eye blink conditioning, that depends on the cerebellum. And so what we have is a biomarker that allows us to reach into the brain non-invasively and see what is happening with cerebellar learning. The technology involves using a phone, programming it to allow things like flash delivery during movie recording, which you can imagine is not what the phone naturally does. Then the responses from the individual being tested then reflect brainstem function. And these responses that depend on cerebellum and brainstem may even depend on cerebrum. And the cerebrum is really the biggest part of the brain and is often thought to be a site of problems in many different disorders. And so this technology is useful not only for autism. There's scientific literature that supports the role of eye blink conditioning as a predictor for autism. It may even indicate cerebellar abnormality that may lead to dyslexia. There's evidence that eye blink conditioning may discriminate Alzheimer's patients. It may be an indicator for Parkinson's disease, and finally for ADHD. And so all these different problems possibly have biomarkers, and we hope that it'll be possible for clinicians or parents or specialists to work with people, kids or adults. And our hope is generally that it will be possible to do these things in the home. I should say that this information is anonymized to protect the identity of people and encoded. And the general hope is that Blink Lab is something that we think will bring smartphone-mediated neurometric screening into everyone's homes. And so the idea is really the brainchild of Henk-Jan Boele, and Henk-Jan and I will be glad to answer your questions. This has been developed by a software team in the Netherlands. We have some pretty high-quality scientific advisors. We have technical staff who've played a critical role. And I should say the early stage development has been enabled in every way by the Princeton Accelerator Fund and key people here at Princeton, Lord Sodikov and Tony Williams. They've really been a wonderful source of advice and also helping us protect the intellectual property. And our early stage investment has been made possible by a partner in Australia, Anton Uvarov. And Anton has been really a great Sherpa in moving us forward. With that, I will stop and I'll be glad to take any questions.
Thank you, Professor Wang, for that exciting presentation. We have time for one question. So could you talk to us about, in terms of timeline, how close the technology is to commercial development and what are the next steps needed?
We think it's going to be a little while. Right now, we are currently doing human testing to build up a database. We've set up operations here in Princeton to do that testing. After that, then that involves setting optimal parameters and then turning it into something that we hope we will start being able to pursue things like questions of reimbursement and classification. I think on a timeline of about, 2 years. Henk-Jan, do you think 2 years?
2 years, yeah. And also want to run a follow-up study where we test young infants and then follow up on these kids. So that will be a longer study that takes about 3-4 years.
Yeah, but I think that we're going to have strong indications about where this technology is headed in less than a year. So we're going to have preliminary information that's going to give a lot of information about the promise of this technology within a year. But getting it licensed and regulatory things and so on, that takes a while.
Perfect. Thank you so much, Sam and Henk-Jan. So this concludes our life sciences track, and up next is our tech track, which will be starting shortly. So please hold on to your seats, and my colleague Chris Wright will be with you in a moment. Thank you so much.